our next speaker, and she is Associate Professor Polly Chen from the Department of Anatomy. Professor Polly Chen is also Principal Investigator of the Cancer Science Institute of Singapore. Her research focuses on functional and mechanistic investigation of RNA in cancer initiation and treatment. She completed her PhD in cancer genetics at the University of Hong Kong and also did her postdoc training in the same lab. So today she'll be telling us, so giving us an overview of her research. And the title of the talk is How Changes in RNA Contribute to Cancer. So over to you, Polly. Just to remind everyone, I think the structure of these talks are roughly 20 to 25 minutes for the research talk, and we try to leave about 5 to 10 minutes for Q&A. Okay, thank you.
Thanks, Jessica, for the very nice introduction. It's really a very good opportunity for me to share my research with all of you. So today I'm going to share our research on understanding how changes in RNA contribute to cancer. So I think it's very a lot of you may know that the DNA mutation can accumulate it in your body that over time, and then they can lead to sorry, I just find my cursors. Okay, so they can lead to cancer progression. For example, the mutations happen at the gene called TP53. It can contribute multiple type of cancer. For example, EGFR mutation will lead to lung cancer, while KRAS mutation can be found in colon cancer and many different type of solid tumors. But now my question is, since you know a central dogma, right? DNA make RNA and RNA make protein. RNA is kind of like the intermediate molecules between the DNA and the protein. So for many years that actually RNA been completely neglected. So we should thanks the COVID-19. Actually, now there are more people aware of how important RNA viruses and how RNA changes contribute to diseases. So my research question is, how about changes in RNA sequence? Does the RNA mutations occur, or whether they can lead to cancer? So I had this question back to maybe 10 years ago. So I was actually a postdoc in my ex-supervisor's lab, and this is a pair of samples from the liver cancer patient. So normal means the tissue adjacent to the liver tumor mass, and tumor means the tumor sample. So we basically collect the tissue samples and extract the DNA and RNA and sample sequencing. So if you just look at this single sequencing data, so basically if you look at the DNA sample in both normal and tumor, you don't see any variation at this particular region. However, when you look at exactly the same samples from the RNA samples from the same patient, you start to see this kind of variation, A to G variation, only happen in the RNA sequence. Then that is the first time I realized, oh, even that DNA mutations there's no DNA mutation, but RNA actually can have some changes. Then I quickly focus on these aspects, and this is how I got to know these RNA editing events. So and after I moved to NUS, so this is my lab. So basically, I was very interested to study how changes in RNA contribute to cancer. So in my lab, there's main research directions. First is more fundamental RNA biology research, which aim to understand the aberrant RNA processing steps in cancer. So as you may know, RNA editing and splicing share the largest overlap in time and space during the processing of pre-messenger RNA and contribute to the transcriptome diversity. So that's why I'm very interested in these two main processes, RNA editing and splicing, and understand how they affect the gene function of cancer-associated genes, and also to understand how they alter the tumor immunity, because currently the cancer immunotherapy is one of the very hot topics in terms of tumor therapies. And besides on the fundamental RNA research, we're also very interested to develop RNA-based therapeutics, but this is actually more recent, only start 4 years ago. So today I mainly pick up those projects that done by my PhD students. So because this is a open house for future potential PhD students in NUS. So the first research question for me, for my lab, is how is RNA editing dysregulated in cancer? So first, I would like to introduce what is RNA editing, because so far I haven't introduced. So basically, in our cells, this is the nuclear. So there's a family of protein called ADAR, ADAR. They can bind to these naturally formed RNA molecules, and then convert this adenosine to inosine, RNA editing. So since these inosine and guanosine share very high structure similarity, so if you do sequencing, you basically will see A to G variation. So if you understand this, let's look at this. So basically, this is also a pair of tissue samples from the same patient. So this is a tumor, this is non-tumor, and this is the DNA. So again, on a DNA sequence, you don't see any variation, but you can start to see these variations in the RNA samples. And you can see their pattern of changes varies among different sites. So some sites showing decrease the frequency, mutation frequency in RNA. However, certain sites showing the opposite pattern of change. So this is what we call RNA editing dysregulation. And I basically spent the first 4 or 5 years on understanding whether this dysregulated editing is implicated in multiple cancer types. Here shows all the published work. Due to the time limit, I don't have time to cover all of this. So basically, these are the cancer types we already confirmed. There is RNA editing dysregulation, which are really contribute to the disease progression. And so I would like to talk about more recent work. So the background and the research gaps in the aspect that I'm very interested in is that it was thought that the factor affecting ADAR levels and activity leading to the editing change. Because as I mentioned, right, so for editing events, ADAR is the upstream regulator. So of course, it's very easy to link that editing change basically is because of the expression of ADAR varies between the tumor and non-tumor. And however, during our research, RNA editing level is not always strictly correlated with the expression and the activity of ADAR enzymes in cells. So bearing these questions, that we actually identify hundreds of ADAR interacting proteins as potential editing regulator. And this editing regulator do not affect the ADAR expression and activity. So which is very different from what other people thought in the area. And these non-ADAR regulators tend to regulate different subset of editing sites via very differential mechanisms. So I would like to give 2 examples on the next slides. So this work are all done by my very talented PhD students. So the first study I would like to highlight is done by my first PhD student, Hui Qi. So basically, she identified the first bidirectional editing regulator in human. So why we mentioned this is a bidirectional means this protein called RNA helicase A, also called DHX9, basically they can either repress or enhance the editing frequency of certain editing sites, and this which has not been reported before. And so at this, about that 1 year later, I had another PhD student, Han Jian. Now he's actually one of the research fellow in my lab, and continue work on RNA editing. So he actually, instead of find another bidirectional regulator, he find out that there's a protein called DAS-associated protein 3, DAP3, function as a repressor of editing, and they can even disrupt. So he provide a lot of mechanism investigation to evidence to show DAP3 can disrupt ADAR1 homodimerization. So if you can see here, DAP3 can inhibit the 2 ADA monomer to form the homodimer, because this homodimer is required for editing reaction to occur. So DAP3 can repress this homodimerization. However, even on the same editing targets, DAP3 can inhibit editing through totally different mechanism by repressing ADA2 protein to bind to the RNA. So you can see that this is a very different from people's understanding that ADA can regulate editing directly without any without additional help from the non-ADA regulators. So this is all very normal findings. And Priyanka, she actually graduated last year. She is more interested in the mechanism of other RNA helicases, for example, DDX6, DDX3X, and DDX21. So so that's why she decided to focus on the regulatory mechanism of these ADA interacting RNA helicase and how they shape the cancer editome. So then you wonder, in our in human cells, there are thousands of editing sites. Which are the sites are related to cancer? So this is a very long-standing question in the area. How you fish out those important editing events among thousands of events. So let me just divide them into, divide this kind of sites into different gene location. The first, we will look at this protein recoding type of RNA editing events. So if the editing sites can be detected in the exon, we call, and they can lead to the amino acid change. So we call this type of editing protein recoding editing events. And they are highly selective and biologically conserved. So meaning that because of the double-stranded RNA structure formation is very unique and in the coding region. And they tend to not form very long, perfect double-stranded RNA structure. So that's why make the ADA binding to the RNA are not so easy to be achieved. So that's why most of the case you only see 1 or 2 editing site within a very long coding region. Okay, so in the past 2 decades, unfortunately, only about 10 protein recoding type of editing events be identified, which are related to cancer. And those highlights are reported by us in the past 5 to 6 years. And currently, we understand these kind of protein recoding editing events can drive tumorigenesis via either augmenting the activity of oncogenes or reducing the activity of tumor suppressors. And very excitingly, that like last year, we on top of this understanding, we actually identified another new mechanism. So I want to just use this diagram to summarize the findings. So COPA is a gene which are involved in cancer development. So what we find is in the normal situation, right? So this COPA protein is edited at this codon 164. So the isoleucine become a violin I2V, okay? So but during the cancer progression, loss of editing make this COPA back to the wild type version, meaning I stay as I at the codon 164. So this mechanism actually turn this tumor suppressive edited version into a tumor promoting version. So now the story is not that simple. So before we thought that editing can make the oncogene more oncogenic, and while repress the tumor, reduce the activity of tumor suppressors. However, now on top of this, we identify a new mechanism. Editing can even switch our tumor suppressor gene to our oncogene, and which leading to the cancer progression. And besides the protein recoding, the exonic region, majority of editing sites are actually identified in the intronic sequence. So what their roles? So because intron, is very important for splicing events, right? Because there are many canonical splice sites, branch points as well as those intronic splicing elements. So it's very easy for us to think that whether there's any crosstalk or interplay between RNA editing and splicing. As I mentioned before, that we are particularly interested in these two RNA processing steps. So that's why my postdoc and another PhD student, so now Chen is also one of my research fellows in the lab. So they spend a lot of time on understanding how RNA editing affect the splicing and vice versa. So what they find is that ADAR can bind and edit the GA-rich intronic sequence. So once you see this kind of GAA, GGAG motifs, these are the region tend to undergo this exon skipping. Okay, so Chen actually also find out that ADAR can affect the circular RNA biogenesis in both directions, which can dependent or independent of their editing function. So all these ADAR regulated linear or circular transcripts can demonstrate higher functional relevance to cancer. Okay. So this is probably the last part of the fundamental RNA research, fundamental RNA biology research I would like to mention today. So I didn't have time to mention that actually a lot of editing occurs in these duplex RNA, because I mentioned that ADAR will bind to the naturally formed the double-stranded RNA structure, right? In our body, we actually have a lot of RNA, double-stranded RNA species or analogs, which tend to be toxic and immunogenic. So without certain immune tolerance mechanism, so you can imagine we may or suffer from the autoimmune disease. So that's why we need certain immune tolerance mechanism to tell our body, okay, this is the self RNA, not non-self. So A to I editing serves as one of these kind of immune tolerance mechanism. So basically ADAR can bind to this endogenous duplex RNA and then mark this RNA as self, okay? Otherwise, it will be recognized as non-self pathogen and trigger the inflammatory and the interferon response. So after being marked by ADAR, so A changed to I, so these are understood as a self RNA and which do not trigger the RNA sensing, meaning they were not recognized by this kind of RNA sensors. So with this mechanism, so for patients suffer from, have this mutation in ADAR protein, so they can cause a disease called Aicardi-Goutieres syndrome. So these patients actually suffer from the spontaneous interferon production in brain tissues. You can see the patients, a lot of spots under the CT imaging. So, and also the mouse data also showing if the mouse have with ADAR1 gene knockout, so they will suffer from a liver disintegration, widespread apoptosis, and upregulated interferon signaling again. So as so because ADAR is frequently upregulated in multiple cancer types, okay, in a normal situation, ADAR can function as a negative immune regulator and serve as an immune tolerance mechanism. However, in a tumor condition, ADAR is overexpressed. And that is the hypothesis that such self-protective mechanism may be hijacked by the tumor cells, ultimately leading to the immune silencing. So this is because, as you may know, that a lot of tumor patients actually have very defective, has defects in the immune protection, meaning they're immune silenced. Okay, so with this hypothesis, actually we already find out there's several papers published in the recent years that loss of ADAR can even overcome the resistance to our very well-known immunotherapy, PD-L1 checkpoint blockade. So that's kind of confirming our hypothesis is correct. So bearing this question, however, in the current area, there are still a lot of scientific gaps. For example, the nature of the RNA molecules with the immune stimulatory capacity are still not known. And also how, for example, the factors and pathways involved in the ADAR-mediated immune suppression are also poorly understood. So my current PhD student, Wei Liang, is currently using generated ADAR1 liver conditional knockout mouse, and together with a very advanced sequencing technology like single cell RNA sequencing, Visium spatial transcriptomics, to understand the factors and pathways which contribute to the ADAR-mediated immune suppression. And due to the time limit, I don't have time to cover all the story. So if you are interested, you can email me after this talk. So this part is just to highlight for my lab, not just fundamental research. We are not just interested in the fundamental RNA research. We are also very interested to translate our basic finding into the clinical applied setting. So due to the time limit, just very quickly went through. So given the role of ADAR and RNA editing in cancer and the innate immunity, so this has emerged as a potential target or process for drug development. So there are many strategies to do so. So you can direct targeting ADAR enzyme. You can also target the ADAR RNA substrates, or you can call ADAR's RNA targets, or you can target the ADAR and RNA binding interface. So this is all different strategy. They all have their limitation and advantages. So for my lab, we are very interested to develop the antisense oligonucleotide platform to inhibit the editing events. And we are also very interested to screen for the very potent small molecule RNA editing inhibitor, but this is a very new research direction in the lab. So I think I only have like 3 minutes left. So just give very basic ideas about what are these platforms. So we have a very established ASO design and evaluation platform. So basically we will in silico predict the RNA secondary structure, and then we will do the in vitro, in cell and cell-free analysis to evaluate the potency of the RNA therapeutics. So the principle is that this ASO can invade into the double-strand RNA structure, which further blocking ADAR binds to the RNA targets. So this is the rationale. And since we already have a very well-developed platform, so there are multiple RNA editing events which are contribute to cancer. You can see tumorigenic, and tumorigenic. So basically using our platform, we can target other cancer-driven events for cancer treatment. Okay, so as I mentioned that we are also very interested in small molecule RNA editing inhibitor development, but since the time limit, so I cannot go deeper. So basically just to share, we have a very well established high throughput screening platform for the small molecule screening. So if you're interested, we can discuss about this off the line. So without further ado, so I would like to, so thanks to all my lab members and all these collaborators at CSI as well as outside CSI. Okay, and this is the funding agency. Thank you so much.